All right, so welcome back to AFF. I believe the last concept that we were dealing with was the oxygen hemoglobin dissociation curve with the partial pressure of oxygen on the x-axis and the percent saturation of oxygen on the hemoglobin itself. We said that that curve tended to look like this because of the cooperative oxygen binding and release of oxygen from hemoglobin. We get these weird shapes at low and high partial pressures of oxygen, and in the middle it tends to be fairly linear. All right, so we had this idea that as oxygen is consumed in particular cells, there should be a buildup of carbon dioxide due to the activity of cellular respiration. If a cell needs more ATP, it needs more oxygen to accomplish the production of that ATP. Coinciding with the consumption of oxygen is the production of carbon dioxide and water. Plus, there's water already present within cells and extracellular spaces. Okay. When CO2 is produced in an aquatic environment, we get carbonic acid. The carbonic acid then dissociates into hydrogen ions and bicarbonate. So this is bicarbonate, and this is carbonic acid. And I mentioned a couple times before, I'll mention it again, the equilibrium continues in natural systems to the production of carbonate, CO3 2-, and 2 hydrogen ions. But this is the dynamic equilibrium as it exists within organisms like us. Okay, so as CO2 accumulates, it drives the dynamic equilibrium to the right, which leads to an increase in hydrogen ions, which corresponds to a decrease in pH. Now, if cells are doing a lot of oxygen consumption, and the consumption of that oxygen is different in different parts of the body. I think I used last time the example of twiddling my thumbs, but the idea here is, right, just make it a little different. If certain parts of the body are under increased metabolic activity, right? We said that thumb there was under increased metabolic activity. That means as blood flows through the circulatory system, oxygen is being consumed at a higher rate here than it is elsewhere within the body. That extra consumption of oxygen leads to an increased production of CO2. An increased production of CO2 leads to an increased production of hydrogen ions, which leads to a decreased pH. Now, hemoglobin reacts to this change in pH by changing its binding affinity for oxygen. So what this curve does is shift down with a decrease in pH. So in our diagram, we have a pH here of 7.4. This curve here is a pH of 7.2. So we've decreased the pH. The hemoglobin moving through the thumb is moving through a lower pH environment because of the accumulation of CO2 in that tissue. The change in the binding affinity is reflected in the change of the function of this oxygen hemoglobin dissociation curve. And what this is saying is that at particular partial pressures of oxygen, for example, this partial pressure, with decreases in pH, you get decreased binding affinity of hemoglobin for oxygen. The consequence of a decreased binding affinity is that the hemoglobin, in the presence of extra hydrogen ions, coughs up the oxygen more easily. That means as the blood with the hemoglobin in it moves through the circulatory system and moves through the body, in regions where pH is low, it coughs up more oxygen, and in regions where the pH is higher, it hangs on to the oxygen a little better. Is that okay? It's almost like this system is designed in such a way to facilitate the delivery of oxygen to tissues that are in need of that oxygen, and to not deliver as much oxygen to tissues that are not currently using oxygen. Is that right? Okay. I have another slide here. It's pretty much the same as the one we've looked at before. It's the shifts in the oxygen hemoglobin dissociation curve. Notice that decreased CO2 and decreased hydrogen ions and increased CO2 and increased hydrogen ions are reflected in the curve, right? A decrease in pH would be an increase in hydrogen ions, leads to decreased binding affinity of hemoglobin for oxygen. But notice also that in addition to CO2 and hydrogen ions, which I would argue are the same effect given this dynamic equilibrium, an increase in CO2 leads to an increase in hydrogen ions. They're also showing temperature here as well. Increased temperature also causes hemoglobin to cough up oxygen more readily. So think about tissue, like muscle tissue that's under heavy metabolic demand, right? You build up heat in those muscles, which should facilitate the disassociation of oxygen from the hemoglobin. So temperature, hydrogen ions, and CO2 have an effect on this curve, but the Bohr shift itself, when we talk about the Bohr shift, we're talking specifically about pH changes affecting the affinity of hemoglobin for oxygen. Okay? All right, so that's the delivery of oxygen to tissues using this pigment, respiratory pigment, that makes oxygen stay in solution better, right? We also need to talk a little bit about kind of the fate of this carbon dioxide in the body when it's produced as a consequence of cellular respiration. So from the dynamic equilibrium here, anytime CO2 is produced, we get changes in hydrogen ion concentration, which changes pH. Well, from a homeostatic perspective, we want to control pH. We need to be able to remove CO2 from the body somehow. Ultimately, we're going to use the respiratory surface to get that, but because we have a circulatory system, we're going to utilize that to move this carbon dioxide from where it's produced in the body to the lungs, where it can be offloaded out of the body. Okay? All right. Okay, so this slide shows us basically what's going to be happening to the CO2 once it's produced in tissues. So at the very top, we have CO2 produced, and all of the CO2 produced should be coming from cellular respiration, right? Specifically the mitochondrion. Krebs cycle is producing CO2, and the preparatory phase, or the bridge reaction, as some people call it, is producing this CO2. The CO2, where it's produced, should be at a higher concentration than in the circulatory system. So specifically, we're talking about here a capillary, right? That fenestrated capillary that allows for gases to move in and out of the capillary system. If it's produced at a higher concentration here, then we should get CO2 moving into the capillaries through diffusion. Okay? As this CO2 moves into the circulatory system, some of it is going to interact with the hemoglobin. Okay? Hemoglobin has the ability to take up CO2, so here's my hemoglobin, remember my quaternary structured protein with 4 separate polypeptides. The oxygen is binding here at these heme groups. CO2 can also bind to hemoglobin, but it binds to specific amino acids on hemoglobin. Okay? Okay, and the amount of CO2 that's bound to the hemoglobin, about 23% of the CO2 that's carried in the circulatory system is actually bound up with the hemoglobin itself at the location of specific amino acids, not at the heme group. And that kind of makes sense. You wouldn't want CO2 binding where oxygen binds, because then CO2 concentrations would interfere with the ability of the heme to bind to oxygen. So here, just like with oxygen, our respiratory pigment is allowing for more oxygen to be carried in this fluid. The hemoglobin is allowing more CO2 to be carried in the fluid by allowing it to bind to these hemoglobin molecules at specific amino acids. So that's this portion here. The CO2 is binding to the hemoglobin itself. There's also that dynamic equilibrium where we're adding CO2 into the circulatory system, and as we add the CO2, we get the formation of carbonic acid and formation of bicarbonate. Right? Bicarbonate. This bicarbonate is shown here. CO2 plus water, carbonic acid dissociating into bicarbonate. The bicarbonate can be carried easily within the circulatory system because it's charged. CO2 doesn't want to be dissolved in solution, but bicarbonate dissolves fairly readily. So the amount of CO2 that's carried in this molecule bicarbonate, that represents about 70% of the carbon dioxide is being carried currently as bicarbonate in your circulatory system. Okay? That bicarbonate can remain within the red blood cells. It could also stay in the fluid, the plasma of the circulatory system, right? Because they're all aquatic environments. Okay? And then finally, there is some CO2 that stays directly as CO2, because after all, this is a dynamic equilibrium. At some point, we reach an equilibrium here with bicarbonate and CO2, and some of the carbon that's remaining as CO2 will be dissolved within the circulatory system. That represents about 7%. Okay, so about 7% of the CO2 in the circulatory system is carried as CO2 directly. Right, so where the CO2 is produced, all of this stuff is happening to get the CO2 into the circulatory system. Now we're at the lungs. There should be a lower concentration of CO2 in the alveoli. CO2 should begin to diffuse into the alveoli. And as the CO2 diffuses out of the circulatory system, we're dropping the amount of CO2 within the circulatory system. As you drop the CO2 from the circulatory system, the dynamic equilibrium should start to move in this direction. It's the reverse equilibrium. Now the bicarbonate goes back to carbonic acid and CO2 and diffuses into the lungs. Here, we're doing the equilibrium backwards. Bicarbonate plus hydrogen ions, carbonic acid gives us water and CO2. The CO2 diffuses into the lungs because of the concentration gradient. As we remove the CO2 should start to come back off of the hemoglobin, right? Back off of the hemoglobin, and as that CO2 is produced again, it moves into the lungs. Okay? All right, so that's pretty much all I want to talk about with gas exchange and CO2 and oxygen. Now what we're going to do is move to the next chapter. And that chapter is digestion. Not a horribly exciting chapter. I think I mentioned last time digestion in all of its glory. It's one of the least interesting to talk about, I think, but it's also one of the more important ones because the digestive system has obvious importance to us as organisms. Okay, now, all of the animals are heterotrophic, which means we don't typically make our own biological molecules, specifically the monomers. What we do is we steal them from organisms that have either made them, like plants, or steal them from other organisms that stole them from plants, like animals. Okay? All right, so let's think about these biological molecules as monomers. And let's start with the first group here. That would be the amino acids. Okay? So there are 20 amino acids that are essential for life. Actually, I'm going to change this to necessary. That will avoid complications with the terminology. There are 20 that are necessary for life so that we can build all of the proteins that we need to be able to build using these monomers. And of the 20, 9 of them, so here let's go 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 of these are referred to as essential. That means we have to have these 9 amino acids in our diet. If we don't have these 9 essential amino acids in our diet, we're in big trouble because we have no way of making these 9 essential amino acids. The others that are listed in this table would be the other 11. These are either non-essential or what's called conditionally essential. More on that in a second.